learn how we produce crops that grow faster, resist diseases, and yield more. One great example is hybrid rice, a breakthrough in agriculture that has helped feed millions. Hybrid rice is developed through genetic engineering, a process where scientists modify an organism's DNA to enhance desired traits. But how does genetic engineering work? Scientists can insert specific genes into an organism's DNA to introduce new functions. For example, by adding a gene from bacteria, plants can gain resistance to pests or diseases. This process allows crops to protect themselves naturally without the need for excessive pesticides.
One example of genetic engineering in agriculture is Bt crops. These crops contain genes from Bacillus thuringiensis, a naturally occurring cyanobacterium that produces proteins toxic to certain insect pests. When these insects consume the protein, it changes shape and binds to receptors in their gut lining, creating holes and ultimately causing their death. However, humans and other animals don't have these specific receptors, so they are unaffected. But genetic engineering isn't just used in plants, it's also widely applied in bacteria. In fact, bacteria have their own natural ways of exchanging genetic material, which allows them to quickly adapt and survive in changing environments. This process is called genetic recombination, and it happens in three main ways: transformation, transduction, and conjugation. Transformation refers to the process by which bacteria take up foreign DNA from the surrounding environment. Transduction is the movement of genes between bacteria by phage. These two processes are between bacteria and closely related species. However, conjugation is encoded by plasmids or transposons. A donor cell attaches to a recipient by a pilus, pulls it closer, and transfers DNA through the mating bridge. Conjugation with plasmid is more common than conjugation with transposons. While bacteria can naturally exchange genetic material, scientists can also manually modify their genes to serve various purposes. By introducing specific DNA sequences, we can give bacteria new traits to study gene function, develop medical treatments, or even produce useful substances like insulin and antibiotics. All of these processes are part of the field of genetic engineering. In our lab, we focused on bacterial transformation by manually recombinating DNA. A key tool in recombinant DNA technology is the vector. Vector facilitates the introduction and replication of foreign DNA within host cells. Plasmid, a circular DNA, is widely used as a vector in molecular cloning, ensuring the propagation of recombinant DNA sequences in living cells. It must contain three essential components: an origin of replication, which enables DNA polymerase and associated proteins to initiate replication, a multiple cloning site, which allows the plasmid to be cut by various restriction enzymes, and a selective marker, which produces a protein that enables transformed bacteria to survive in selective conditions. So what we will do in this lab is, firstly, introduce PUC19 plasmid into E. coli DHA5A competent cell. Secondly, plate and incubate bacteria and observe colonies. The PUC19 plasmid carries an origin of replication an ampicillin-resistant gene, and the LAC-Z gene. The LAC-Z gene encodes the beta-galactosidase enzyme, which converts the X-gol into a colored product. Thus, the bacteria colonies would exhibit blue. This sheet lists all the materials required for this lab. Here is the procedure. Label 2 tubes and 4 agar plates. Second, after thawing the competent cell, transfer the cells to 2 tubes. Third, add plasmid solution to 1 tube and apply the heat shock method. Fourth, add LB medium to facilitate recovery from heat shock. Fifth, incubate the tubes at 37 Celsius for 20 minutes. After spreading solution onto corresponding agar plates, incubate agar plates upside down at 37 Celsius overnight. This is what I got on the following day. We can find that PUC19 transformants grew as evenly distributed blue colonies on the agar plate with ampicillin. On the agar plate without ampicillin, PUC19 transformants formed white colonies, and although there may have some blue colonies, they were not visible due to the dense growth of the white colonies. In the case of PUC19 non-transformants, the agar plate without ampicillin showed a white bacterial lawn, as expected. However, the agar plate with ampicillin showed no bacterial growth, indicating that the non-transformed cells could not survive without the ampicillin resistance gene. Well, what are some real-world applications? Diabetes is a chronic condition that affects how the body regulates blood sugar or glucose, which is our main source of energy. When blood sugar levels are too high or too low, it can lead to serious health complications. There are 2 main types of diabetes. Type 2 diabetes is very common in our daily life and usually develops over time. It is often linked to lifestyle factors such as diet, exercise, and genetics, and it can sometimes be managed through lifestyle changes and medications. However, type 1 diabetes is an autoimmune disease where the body's immune system mistakenly attacks the insulin-producing cells in the pancreas. As a result, the body produces little to no insulin. People with type 1 diabetes must take insulin injections or use insulin pumps to regulate their blood sugar. The extra insulin allows the glucose to be taken up by the liver and other tissues, so cells get the glucose they need and blood glucose levels stay normal. Natural insulin can be taken from the pancreas of pigs or cattle. However, this insulin causes adverse reactions in some people, and its supply is limited. Therefore, nowadays, most insulin is made using genetically modified bacteria that have had the human gene for insulin inserted in them. This genetically modified insulin has some advantages over insulin taken from pigs or cattle. Firstly, it can be made in very large amounts from bacteria grown in a fermenter. Secondly, it is less likely to cause an adverse reaction. Thirdly, it overcomes ethical concerns from vegetarians and some religious groups. There is a gene that codes for making the protein insulin in most of our body cells. This insulin gene can be inserted into a bacterial plasmid. This plasmid can replicate continuously. Thus, the bacteria can use the human insulin gene to produce human insulin and the insulin can be purified in the lab setting to be used in humans with diabetes. Bacterial transformation through genetic engineering has revolutionized various industries, including medicine, agriculture, and biotechnology. This technology has provided groundbreaking solutions to global challenges. However, its use also raises ethical concerns. Questions about biosafety, antibiotic resistance, and unintended ecological effects must be carefully considered. With the development of genetic engineering, it's crucial to ensure that these advancements are used safely and ethically for the benefit of society. At the end of this video, I would like to express my sincere gratitude to our amazing professor, Chun-Hyun Lee, for his guidance and support throughout this course. I am also deeply thankful to our dedicated lab instructors, Dr. Su-Hua Lee and Dr. Jing Miao, for their invaluable assistance during the lab session. Their encouragement and insights have made this learning experience truly enriching. Thank you all for watching, and see you next time.